Our company started about 5 years ago, at first in close association with my home institute at that time, the Rockefeller Neuroscience Institute, which arose out of the invitation of the Rockefellers to start a new neuroscience institute focused on Alzheimer's disease. I had, with my teams at NIH, been working on molecular mechanisms of memory. At that time, we looked at the possibility of bringing that work to the clinic, and it was obvious to us that we should start with a disease that is known paradigmatically to start with recent memory loss. It doesn't always start with that, but there is no other dementia that actually can start with recent memory loss. And we moved some of our molecular mechanisms of memory storage to their relevance to Alzheimer's disease. That research, which actually continued from the NIH into the Rockefeller Institute, eventually spawned a therapeutic and diagnostic approach. The therapeutic approach actually involved synaptogenic pathways that were actually governed by PKCϵ, a target which coordinates a variety of synaptic growth factors to generate new pathways, and as well to prevent neuronal death. At that time, we didn't know, but we hypothesized that the benefits of such an approach could span a variety of neurologic conditions that involve the loss of the wiring or the synaptic networks in the brain. The degeneration could be caused by, a gene. It could be caused by a gene for Alzheimer's or a gene for Fragile X. It could be caused by stroke, traumatic brain injury. In all of these cases, the consequence is the loss of the synaptic networks, analogous to the wiring in a computer. If you reach into a computer and rip out the wiring, you're not going to have much of a function generated by the computer. Similarly, if you have a consequence, whether it's Fragile X because of a gene even early in childhood, or whether it's Rett syndrome, or whether it's Alzheimer's disease, which our company Neurotrope is focused on right now, in all cases, you have a daunting task. How can you regenerate those synaptic networks? We believe that Neurotrope has a unique and new approach to replacing the synaptic networks. We have a platform of drugs led by a compound called Bryostatin, which is not a statin, which can be administered to patients and then cause impact potentially sufficient to actually reverse symptoms of loss. And in our preclinical studies, we not only focused on Alzheimer's, but we also focused on Fragile X. And in fact, the work that we did in Fragile X on both young models and older models was sufficiently exciting. It actually was validated with electron microscopy reconstruction to show that the networks were being replaced and the cognitive functions of the animals were being replaced, that the Fragile X Foundation asked whether they could repeat our study in Chile in their laboratories, which they did. And they not only replicated what we had gotten independently, but they also extended the findings to autistic spectrum disorder. So they really provided a great deal of validation of what we were doing in animal models. Sufficient that when we went to the FDA asking for orphan drug status, the FDA granted it within a matter of weeks. So we have orphan drug status for Fragile X, which is somewhat of an accelerated path to clinical trials. And that has given us the impetus to plan clinical trials for Fragile X sometime in 2018. We expect to be working with a collaboration program where experts in Fragile X and clinical and other aspects would be collaborating with us. That is a philosophy that Neurotrope has extended to not only Fragile X, but to other potential indications. So we have, we believe, a platform approach that has a couple of different dimensions. One dimension is spanning different indications, whether it's Alzheimer's disease, which is our major focus and which we're very excited about our latest phase 2 trial data, showing actually an improvement in the patient, very severe improvement in the patients that we could quantify. but also to a variety of other indications, such as stroke, Fragile X, traumatic brain injury. And the other way of looking at the platform is that we don't just have Bryostatin, which is our lead compound. We have 40 other compounds, many of which we synthesize and basically constructed ourselves. So we have the potential of follow-on new chemical entities that can be on a path for higher order generation drug approaches.